All right, so I need to also do this second example for you. So first I need G fragments. They would look like this: ACG, AUCG, and then a C. Whereas my UC fragments, I'd break here, 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 here if I needed to. Again, AC, GAU, a C, and then a GC. So then I go in the G fragments, hyphens behind U's and C's. This one actually has two hyphens because it's got a U and a C in it. This C goes nowhere, so we're probably not going to use it. But again, the overlap connects these two groups together. I use hyphens behind G's in my UC fragments. If there is no G, then I don't use it. Otherwise, when it's all said and done, I have to make a path, and I'm going to start with this first one here. I'm going to go from AC to AG. Doesn't really matter where you put those things as long as it's organized and you can connect, right? This next one is the weird one, though. If you have two hyphens, you do something different. This one goes from an AU to a G. So I have the G. I'm going to do an AU to a G, but I also had that C in between those two hyphens. I just put that on this line, traveling from AU to G, and that's how you deal with the extra letters here and there. So I have those two done. I also need to go from a G to an AU. Those exist already, so I just connect, and then G to C. Now, when I say G to C, I can't use that C that's on the line, so I have to add a new node somewhere, right, for my path to follow. So it ends up looking like this. When it's all said and done, I need to travel I have to go along from each node following each path once. You should also be able to recognize that I would start with an AC and I'd probably finish over here with a C. But as I follow, I just write down letters. So AC goes to G, follow that path. Now I take the G to the AU, and then I go back to the G, but I put that C first because I pick up any hitchhiker that's on the path on the road from AU to G. And then finally G goes to C, so that should be my completed piece of mRNA here that looks the same as this RNA up here. Again, technically on the real work that you do, you don't get to see what this original RNA looks like. So you have to come up with a sequence, and this is how we sequence RNA. And that's important because ultimately that's what we do to figure out what our DNA says. Remember, it would actually be kind of pointless to code to figure out the sequence of the entire chromosome, right? Each one of my 46 chromosomes, because 98% of that chromosome is random gibberish, right? So I don't need to know what it says. I only need to know what those genes say. And every gene, if it's expressed, is expressed through RNA. So that's how this works. That's why we're doing this. So we isolate RNA out of cells and we code that. We figure out the sequence for that RNA, and then we could reverse figure out what the sequence of the DNA would be, and then we know what all our genes are, right? So that's that.